I have the pleasure today of interviewing Dr. Vic Nanda, who is one of the faculty members at CABM. Vic, welcome. So Vic, tell us about your education and your early career before you got to CABM.
Okay, so I've always wanted to be a scientist ever since I was a little kid, and I used to love doing science fair projects. And so when I had the opportunity to go to college, I went to a place which was very science focused. I went to Caltech in the early 90s, and there I majored in biology. After college, I came back to the East Coast and did my PhD at Johns Hopkins in biochemistry. And there I worked with Dr. Lenny Brand, who's still there. And in his laboratory, we did optical spectroscopy of proteins. But also while I was there, I had the opportunity to play a lot with very high-end computers that could look at the structures of proteins and model their dynamics. And so I became very interested in both doing experiments in the lab and running calculations on computers. And then after that, I did my postdoc at the University of Pennsylvania in the Department of Molecular Biophysics with Bill DeGrotto. And there I became almost fully computational, writing software to model proteins. And this piece of software allows you not only to predict the structure of proteins, but also to engineer them, to introduce mutations and change them. And
Great. So how long have you been at CABM, and what brought you here in particular?
Well, I was hired in 2005. So I've been here now for 15 years. And, CABM, at the time that I interviewed and since then is a very special place for a number of reasons. One is that it's very interdisciplinary. And I like the type of work that we do when studying the structure of proteins lends itself to collaborations with a lot of other fields, from neuroscience to genetics to virology, because a lot of them have proteins as crucial components, in their study. And really sort of what combines all of the different projects that we're working on is trying to understand why the proteins have the shapes that they do. And in understanding the evolution of these proteins, we can then understand the basic principles that control their shapes. And then we as engineers can then use that to make proteins that can function as therapeutics or can function as catalysts, these sorts of things. So really that's what sort of drives our science.
Yeah, really interesting. So what are some of the approaches you're using to address this issue of protein shape and protein evolution?
Sure. So, I think one of the key technologies that has really moved not just my research, but the whole field of protein science forward is the availability of large data sets of protein structures, the atomic resolution three-dimensional structures of proteins. And this resource is at Rutgers University at the PDB, which stands for the Protein Data Bank. And currently there's about 150,000 protein structures in the PDB. And so one of the key ways that we study this is by writing software or running calculations that try and take that large data set and reduce it to elements that we can work with. We're well aware that our current understanding of the chemistry and the physics that controls the shapes of proteins is still lacking. And so whenever we use computers to make predictions, we then need to go into the laboratory, synthesize these proteins, express them in cells, test their activity, and see how well these predictions bear out.
Yeah.
That's kind of an iterative cycle. We make a prediction on the computer, we test it in the laboratory. More than often we're wrong, and then we go back and we change the software, and it sort of is is a way for us to sort of gain that sort of basic understanding.
Yeah, great. So with this in mind, can you tell us about one project in your lab that you're particularly excited about?
I love all the projects in my lab, but one that we're having a lot of fun with right now that I'm particularly excited about is we were a few years back we were funded by NASA to study astrobiology, and it really is an aspirational goal to sort of imagine what life would look like on other planets, right? And in order to understand what life may look like on other planets, we need to understand how did life evolve on Earth. And the problem with understanding other planets is that they're far away. They're light years, if not, millions of light years away. And so we're limited by what we can see in the telescope. And then for Earth, we're on Earth, we can see life as it is today, but we're limited because we can't see what life looked like billions of years ago. What we're doing right now in the laboratory is trying to reinvent the origin of proteins. And we're doing this by taking that database of protein structures and looking at it to see if we can identify fossils inside of those proteins. And the proteins we have today are huge. They're massive nanomachines that are very complex. But inside of these proteins, we think are smaller, simpler parts, the engines, the wheels, the axles, and so forth, the transmission. And these were the parts that probably were there on the planet early on. And what we're finding, this is, to me, Marty, this is the most exciting part. What we're finding is that these small peptides that we're making, not only can they do the functions of the larger proteins that they came from, but sometimes they even do them better. Sometimes they're stronger, their activity is higher, they're more chemically stable. And this has been for us a very interesting paradigm shift, because we sort of think of evolution as taking things and making them better over time. But what we're finding is that, the earliest molecules were actually quite good. And so this is something that's very exciting to us. And we've been trying now to do this for a number of basic proteins that we imagine must have been there early in Earth's, in life's evolution.
Of course, they may be better now because they have more regulation. The others were a much simpler system, whereas the improvement is that they're more regulated and they're more context dependent, for example. That's what I would guess.
Absolutely. I mean, I think that what is, when 4 billion years ago, if you were the first protein and you were, huddling on the surface of a rock in the ocean and it was being bombarded by UV radiation, you didn't care about, interacting with other proteins in the cell. You just needed to survive. But I think over time, what evolution has done is it's added all this bureaucracy to proteins. Now, proteins have to interact with other proteins inside of a cell. They have to be turned on at a particular time. They have to be broken down quickly because you may not want the protein lingering in the cell. And so I think a lot of that complexity is, as you say, it has to do with regulation. Absolutely.
So then I can quote you in saying that bureaucracy is necessary and advantageous.
Well, certainly from evolution, right? It's been the easiest path for evolution. Yeah.
So that sounds like your lab's a really exciting place. Can you tell us how you interact with students and other trainees?
Sure. So, I think that one of the exciting things about working in an area like astrobiology is that it is incredibly interdisciplinary. And the way that we've set up this particular project, I am one PI on this project, but there are several. And they span different fields, from environmental science to geology to bioinformatics. And when we bring in graduate students and when we bring in postdocs, they don't belong to any one particular lab. They actually belong to the project as a whole. And they spend time working in all these different groups. And that allows us to do a different kind of science, right? And it allows us to do things that bridge a lot of different technologies and a lot of different ways of thinking. But it also, I think, gives the students themselves critical training in networking, in working, in larger teams. And also, I think that one of the important things, science is a very hard field to be in, because most of the things you do don't work, right? Most of the things you do, end up, not coming out the way you expect. They end up failing. And it's important to remember that science also needs to be fun, right? And if you lose sight of that fun, then it can be very discouraging. And so that's something that I constantly try to remind people, is that when you're working on a project, you may be really focused on your particular problem. But there are these bigger questions, that we're trying to answer. And just because something doesn't work doesn't mean that we've lost sight of that question. And you got to remember that those are the things that are really why we're coming to the lab every single day to do this sort of work.
Great. Very well said. Well, Vic, thank you so much. What an interesting interview. Thank you.
Marty, I appreciate it. Thanks for your interest.